Hello everyone, this is Prashant and we are starting with our second topic. In the earlier video, we discussed the questions that will lead us to a new pathways. Okay, what are brain? What are their functions? What are nerves? How do they carry? What do they carry and how do they interact with the brain parts and other body parts? Okay, so we are going to kind of integrating all that data or answer all those questions. Okay, so we are starting with what? Structure of the brain. Okay, earlier we know that Versalius kind of discovered and mapped out the whole brain system and nervous system. And here we are going to specifically uh, discover each brain part and their function. Okay, so like a child, what we are going to do is we are going to observe and investigate each brain part. We are just going to do it in different forms, in different positions, and we are going to map out, okay, what each part is, what is their name, and where are they located. Okay, that's what we are going to do in this video. So, yep, let's start with our neuroanatomy. So, okay, we know that brain is an organ that resides within our skull, you know, at the top of the body, inside our head. Okay, based on its structure, and we can observe that brain exists in three-dimensional world, so it can be observed in three planes: x-axis, y-axis, and z-axis. Okay, obviously, when I take out the brain and cut it in different planes, I get the following diagrams. Okay, from different places. Okay, that is observable and self-evident. Okay, so what do I observe? Okay, first of all, that brain is like a gooey structure, and it has what you call it, lobes. Basically, there are big divisions inside across your brain, and basically, brain can be divided into four lobes or four foldings. Okay, structurally, and when you cut to the brain, I realized that there is a space between the brain. For example, there are kind of literal blank spaces within the brain, and there is some form of fluid in it. Okay, so there are four lobes: frontal, parietal, occipital, and temporal, and they are what you call it, ventricles, that are the empty spaces between your two brain hemispheres. Yep, brain is symmetrical. So yeah, those are our observations based on the brain structure. Okay, so obviously, from different positions, I label different parts. Obviously, I can observe lobes from the topmost, and then there are other parts like cerebellum, occipital, cranial nerves, and yep. So basically, I just slice the brain in different parts, and I name each part. Okay, those are that on the outside and the inside. Okay, so overall, this is the summarized version of all the brain parts, main brain parts, and how they are kind of labeled or named. Okay, you can see it. We start from the bottom, like from the brainstem, and then we move forward to the cortex. Okay, so yeah, this is observable as well. And this was the ventricle I was talking about, the empty space between two brain hemispheres. And there are white matter, gray matter, which is kind of literally uh the different types of concentration of tissues in your brain. Yeah, so there's that. Okay, there are cerebral ventricles as well. Okay. So basically, what do they do? They carry cerebrospinal fluid. Okay. What is the main function of that fluid? Okay. So there are many main functions. Most probably, it are those that it helps to cool down the brain. It supplies nutrients. Uh, it's more like drainage system. It also carries out the waste that the neurons produce in your brain. Okay. We are going to discover what neurons are later, but I'm just giving you a what you call it, a preview of what we are going to cover. Okay. And then I also observed that brain is kind of surrounded by different arteries that carry blood. And the main function of the blood is to provide nutrients and oxygen to different body parts. Okay, cool. So there are three main types of arteries that surround the brain. Okay, now that we have covered it, we are gonna move forward to the functions of the brain. Now that we know that, okay, this is the brain structure and how we label the brain structure. we are gonna move forward to the functions of each brain part. Okay, so let's start with our first scientist, that is Joseph Gall. So what did he discover? Okay, his method was known as phrenology. So what he did, basically he made a list of all the mental functions, physical functions that humans perform, and then he took out skulls, not brains, of different people, and he started to observe them, either of dead people or living people. He just started to observe, okay, what is the shape of their head, how one shape of head is different from other. He kind of and he started correlating, okay, suppose if people from having a certain head size showed certain attribute, like people with big head scored better marks or worked in a higher level positions, he kind of correlated it with intelligence, something like that. Okay, so he made these broad correlations between different skull, what do you call it, size of the skull and different parts of the skull, and then he started to, what do you call it, labeling them as uh that part associated with certain function. Okay, so his reasoning was as follows. He was like, okay, now we know that brain is associated with sensation and mental functions. So scale, so there are bumps on the skull or unevenness on the skull. It must be due to certain brain development of certain brain parts. And we know that people have different brain sizes, skull sizes. And we also know that brain is associated with mental and physical functions. So he was like, okay, so instead of studying the brain, he studied the skull and he made broad correlations that, okay, certain form of skull leads to certain form of mental and physical functions. Okay. And this was known as phrenology. He was localized the brain functions. Basically, he said, okay, in certain part of the skull or head, certain functions are performed by the brain. Obviously, he didn't exactly specify which brain part. He was uh, on the outer level. He didn't go inside or dissected the brain or try to do any form of experiments. He made broad correlations. Okay. Obviously, this approach was not correct, so we came up with a new scientist whose name was Pierre Florence. So, what was his method? Okay, he conducted actual experiments on animals, and in his experiments, what he did was he did ablimation, which is he destroyed certain brain parts and observed the effects to other animals whose brain parts were intact. So he had one experimental group and one control group. You can say that. And he refuted the phrenological correlations. And how did he refuted them? He found out that, okay, the map that phrenologists gave of the skull size did not match the actual experiments conducted by ablimation. Okay, and experiments by ablation was more, what you call it, consistent and reliable and valid. So let's start with his reasoning. So his reasoning was as follows. Okay, when I remove cerebellum, it causes problem in motor coordination. When cortex is removed, it causes loss of will and problems in sensory perception. When medulla oblongata is removed, it causes the death of the animal. Okay, so damage to specific part causes specific behavioral dysfunction. Okay, so certain brain parts are responsible for certain functions, not the skull sizes of the organisms. Okay, that was his main conclusion. And he was unable to locate any cognitive functions, high level cognitive functions, obviously, because he was experimenting on animals. He was not experimenting or observing humans. So he was not able to, what you call it, pinpoint the location of certain cognitive functions like language or thinking, which is not possible in animals. It can only be observed in humans. That what separates humans from animals. So obviously, now that we have experimented on animals and we found out that certain brain parts are associated with certain physiological or mental functions, we are gonna go start the localization of mental functions. Okay, so we are gonna come to humans, and that is we come to our scientist Paul Broca. So what did he discovered? So through his method, That is, he observed patients who had speech problems. Basically, they could not produce any form of speech, they could not talk. So he conducted autopsies when those patients were dead, and he found out that certain brain part was damaged. Okay, this was clinical method. This is after the patient was confirmed having a certain problem, diagnosed with a certain problem. He was dead, and then autopsy on him was done, and his brain part or brain parts were observed, and he found out that left frontal lobe of cortex was damaged, and this led him to certain line of reasoning, and that reasoning was specific brain parts are responsible for specific functions. We proved that experimentally, and damage to those brain parts causes dysfunctions in or certain types of dysfunctions. Okay, and we also found that aphasic patients who cannot speak, have damage in their left frontal lobe. Okay, so we can kind of form a causation that left frontal lobe region is responsible for higher cognitive function of speech or language. Okay, and yeah, this means higher cognitive functions are also localized in the brain, which means you can actually pinpoint and okay and find out that this brain part is responsible for this form of function. And we used to call it Broca's region. Obviously, there was Wernicke's region as well, and so on and so forth. And this process was repeated over and over again. Okay, where certain certain brain part was damaged or certain damage in a brain part was observed, and uh, cause and effect was formed between brain and behavior. Okay, so we are coming up to our next scientist, and there are two scientists here, Edward Hitzing and Gustav Fritsch. So what did they find out? Okay, their method was to do electrical stimulation of brain. Why? Because from Galvani's research, which we are going to discuss later on, which found that brain consists of neurons, and neurons are electrochemical in nature. Okay, and they are affected by electricity. So what did they observe? They observed that soldiers who were injured and had head injury, when you actually stimulate their brain through electricity, it was called involuntary motor movements. Basically, if you stimulate certain parts of the brain, it will cause movement in your body. Okay, and they use that electrical stimulation to pinpoint which specific area of brain led to what form of movement. Okay, so their reasoning was as follows: nervous system is electrical in nature. We know that from Galvani's research and other people's research, and brain functions are localized. We know also verified that. And then what we found out that left hemisphere stimulation led to movement in the right body parts on the right side of the body, and vice versa. And this led to the discovery of cortex. Basically, it's a kind of region around your lobes, you know, on your frontal lobe. And when you stimulate that specific brain parts, it will help you discover particular motor functions. Okay, so electrically stimulating brain also leads to what you call it discovering what kind of brain functions happen. So in this way, we don't have to actually damage the brain itself. We can actually observe and control brain with electricity and without actually damaging the patient in the long term. So yeah, so now we don't have to actually what you call it experiment on the dead people. We can actually experiment on living beings as well. So this leads us to our new researcher, and his name was Wilder Penfield. So what was his method? His method was electrical stimulation uh, and surgery procedures. He discovered new surgery procedures. And what did he found out? He observed that certain brain parts, when stimulated, causes sensations in different body. Earlier we were discovering movements generated in the body. Now we are kind of what you call it observing what kind of sensations are generated in the body, and they localized its function. So they found out, okay, when the green part, okay, uh, which is also known as somatosensory cortex, when it is stimulated, it causes stimulations in different body parts on your eye, neck, nose, ear, something like that. So their reasoning was this. Brain functions are localized in brain parts. Okay, connections are found through electrical stimulation, as done by the experiments of the previous two scientists. And left hemisphere stimulation leads to sensation in the right part. Okay, and this led to the sensory discovery of somatosensory cortex. Okay, and certain form of stimulation in left temporal lobes also causes recall of memories. For example, if your temporal lobe is kind of stimulated, we found out that you can recall certain memories or you get certain form of flashbacks or visions. Okay, so this was his discovery. Okay, now that we know that brain is associated with certain physiological functions, mental functions, okay, and higher cognitive functions, we can move forward to a new researcher whose name was Roger Wolcott Sperry. And what did he discovered? Okay, so his method was to find the effect of removing corpus callosum, which are kind of fibers that connect to brain hemispheres, and how do they affect the brain function. So he did multiple experiments on humans or animals, okay, through lesion or through uh, electrical stimulation. So one experiment he did, that is the main one. Okay, so he presented two separate images for each eye. Okay, left eye is connected to the right hemisphere, and right eye is connected to the left hemisphere through connections of neurons. Okay, and he asked the subjects to describe what kind of image they saw, and he made the setup in such a way that one eye can only see one image and the other eye can see only other. Okay, so what did he discover? That the right eye was able, that was connected to the left hemisphere, was unable to observe what the left eye observed. So what did he find out? He found out that left hemisphere is able to process speech, but right one is not. So how? Okay, what he saw with the right eye, and we also know that left frontal cortex, what you call it, left frontal cortex of Broca regions is it related to speech production. Uh, is on the left hemisphere, and it cannot observe, what you call it. It can produce speech, but right hemisphere cannot. He was like, okay. So each hemisphere is dominant in certain form function. So left hemisphere is dominant in speech production, and right hemisphere may be associated with other functions. So after doing a lot of experiments, they kind of divided brain into left brain and right brain. Okay. So we came up with the left brain and right brain analogy. So his reasoning was this. Each hemisphere is connected to the opposite side of the body. Okay. Makes sense. And each hemisphere has certain functions that the other one does not. Like left hemisphere can produce speech, meanwhile right one cannot, which leads us to lateralization of brain functions. Okay, like each brain part controls a specific function, not entire brain. Okay, makes sense. And the function of corpus callosum is to connect brain parts, because if your brain parts are disconnected, you may not be able to function properly. You can only do things half correct at a time. Okay. So he found out that left hemisphere controls speech comprehension, arithmetic writing. Meanwhile, right hemisphere is associated with creativity, spatial ability, artistic and musical skills. Okay. So that led us to the discovery of left brain and right brain. Uh, so that's what we discovered. I hope I'm clear. Okay. So that's all. And in the next video, we are going to I'm going to summarize all the results, all the conclusions of all the scientists, okay, about which brain part is associated with which function and why. And that's it. So thanks for watching. If you like the video, maybe give maybe give it a like, maybe comment, maybe subscribe, share it if you find it useful. So yep, I will take my leave, and thanks for watching and good luck.